Welcome to Part 3 of the Lecture 13 on plant hormones. Please ensure that you have watched Part 1 and Part 2 of Lecture 13 before continuing with Part 3. This lecture is part of the plant physiology subject, which is a component of the agricultural and viticultural degree programs offered at NMIT. Please refer to the recommended reading from Tays and Zeiger, Chapter 19 to 24, for the complementary information. My name is Dr. Nikki Cooley. In this lecture, we are going to concentrate on three hormone groups: the gas ethylene, abscisic acid or ABA, and finally, completing this series on plant hormones, the baracinosteroids. So let us begin with the plant hormone ethylene. Ethylene's molecular composition is 2 carbons and 4 hydrogens, and the model on the screen shows its chemical orientations. Ethylene is a gas. During the 19th century, when cool gas was used in street lamps, it was observed that nearby trees defoliated more extensively. In 1901, a botany student in Russia observed that dark-grown peas exhibited symptoms later called the triple response: reduced stem elongation, increased lateral growth, and abnormal horizontal growth. Ethylene from cool gas in the lab was responsible. Almost all parts of the plant can produce ethylene. Production increases during leaf abscission, flower senescence, fruit ripening, wounding and physiological stress, including flooding, disease, temperature and drought stress. The amino acid methinone is the precursor of ethylene. The major enzymes in this pathway are ACC synthase and ACC oxidase. The ethylene biosynthesis starts with induction of ethylene biosynthesis. The figure on the screen illustrates some of our understanding to date into the insights of this complicated process. You will not be required to remember this process, but only appreciate the complications. After ethylene biosynthesis has occurred, downstream ethylene sensing and signaling pathways occur. Again, these are shown on the slide. In a recent paper published in 2006, ethylene biosynthesis in rice was illustrated. For the level of this subject, you will not need to know the intricacies of this pathway, only that there are many steps and that there are many intermediates. Ethylene is the hormone that promotes ripening in some fruit. Ripening is the softening due to enzyme breakdown of the cell walls, starch hydrolysis, starch accumulation, disappearance of some acids and phenolics, including the group tannins. Dispersal by ingestion is where ripening also involves the accumulation of brightly coloured anthocyanins or carotenoids. Ripening means that the seeds are ready for dispersal. Fruits that ripen in response to ethylene exhibit a respiratory rise called a climeric before ripening. The figure on the slide in banana illustrates ripening characterized by climeric rise in respiration of carbon dioxide, which precedes by an increase of ethylene. Fruits can be separated into 2 groups, those that are climeric and those that aren't. The table on the slide shows you some examples of each of these groups. Ethylene induces root growth. It has been shown that ethylene can induce advantageous root formation in leaves, stems, flower stems and other roots. For example, vegetative stem cuttings from tomato and petuna. Ethylene also promotes root hair formation, as shown on the illustration in the slide, in lettuce seedlings. Ethylene is a hormone that can enhance leaf senescence. For example, exogenesis application of ethylene accelerates senescence. Inhibitors of ethylene retard leaf and flower senescence. Carnations in silver thiosulfate, or STS for short, is an inhibitor of ethylene action, and it results in inhibition of senescence. Again, understanding the function and mode of action of ethylene in plants has been very useful from the terms of the commercial agricultural perspective. Ethylene is one of the most widely used hormones in agriculture. Ephron, discovered in 1960s, is sprayed in solution to hasten the ripening of apple and tomato. It synchronized flowering and fruit set in pineapple. It induced fruit thinning in cotton, cherry and walnut. It is responsible for degreening of citrus fruits. It prevents self-pollination and increases yield, and it can promote lateral growth. The ethylene inhibitor, ethyl block, is used to extend the shelf life of various fruits. The next hormone we are going to cover in this lecture is abscisic acid, or ABA. On the slide you will see a molecular structure and a modelled structure by the Nutritional Immunology and Molecular Medicine Laboratory. ABA is a phytohormone that serves as the prime signal in response of plants to environmental stresses imposed by cold, drought and high levels of salt, as stated by Christemin et al. In their 2006 paper. ABA was originally discovered in leaves from sycamore trees and named Dorman. ABA is an important plant hormone that regulates growth and stomata closure, seed maturation and dormancy. ABA is synthesized in the chloroplasts and the other plastids via the terpenoid pathway. ABA is transported via the xylem and the phloem, but is much more abundant in the phloem. ABA biosynthesis, like many of the hormone pathways, is complicated and intricate, and it has many intermediates. From the perspective of this subject, you only need to understand that it has many intermediates. ABA is one of the components that is responsible for the closure of stomata. ABA concentrations in leaves can increase up to 50-fold times under drought conditions. Redistribution of ABA is very effective in causing stomata to close. Its accumulation in water-stressed leaves plays an important role in the reduction of water loss through transpiration. On the figure on the slide, produced by Christemin and Gill in their 2009 paper on ABA, they demonstrate how the compound ABA is involved in the opening and closing. On the right-hand side of the picture are some of the genetic pathways involved in this process. Let us examine the process of ABA and stomata closure in more detail. ABA binds to receptors. The ABA binding induces the formation of reactive oxygen species, or ROS, and this in turn activates calcium channels on the plasma membrane. The influence of the calcium interacts with intercellular calcium transients and promotes further release of calcium from the vacuoles. ABA stimulate no production and no increases of the CADPR levels. ABA does increase the INSP3 levels via a signaling pathway that induces S1P, heterotrimeric G proteins, and phospholipidase C and D. The rise in the CADPR and INSP3 activates additional calcium channels on the tonoplast, releasing more calcium from the vacuoles. The rise in the intracellular calcium blocks potassium channels on the plasma membrane. The rise in the intracellular calcium promotes the opening of chlorine ion channels on the plasma membrane, causing depolarization. The plasma membrane protein pump is inhibited by the ABA-induced increase in cytosolic calcium and a rise in the intracellular pH, further depolarizing the membrane. Membrane depolarization activates potassium plus ions out of the channels and onto the plasma membrane. Potassium ions, to be released across the plasma membrane, are released first from the vacuoles in the cytosol. In summary, this process is an elegant process that involves not only hormones signaling calcium and potassium and chloride channels, and ATP in the form of energy. Stomata close due to the net effect of the loss of potassium ions, chloride ions, and the subsequent osmotic loss of water. The surface area of the guard cell plasma membrane might contract by as much as 50%. The loss of turgor induces closure of stomata to prevent further water loss by evaporation. And this brings us to the last group of hormones we are going to study in this lecture, the brasinosteroids. On the screen you will see a typical structure of a brasinosteroid. There are also images of Arabidopsis and mutations involving brasinosteroids. The brasinosteroids, or BR for short, are a group of steroid hormones that play a role in cell division and cell elongation in stems and roots, reproductive development, leaf senescence, and some stress responses. The early studies in the 1960s showed that the greatest growth stimulating activity was found in extracts of pollen from rape plant, shown to increase total biomass and seed yield. Therefore, the US Department of Agriculture provided funding for laboratories to purify and identify the compound. Approximately 60 related brasinosteroids have since been isolated. They are synthesized via the terpenoid pathway. In a publication in the Brazilian Journal of Plant Physiology in 2002, a review article was written on the brasinosteroid pathway. The pathway in that review is presented on the slide. As with all other biosynthesis of plant hormones, this pathway is complex and involves many intermediate steps. Small-scale studies have shown that brasinosteroids can increase bean crop yield by 45%, enhance leaf weight of lettuce by 25%, increase yield of rice, barley, wheat and lentils, promote potato tuber growth, increase resistance to infections, and enhance tomato fruit set. While large-scale studies in Japan, China, Korea, Russia have shown variable results, minimal effect on crops grown under optimal conditions, or dramatic effects on crops grown under stress conditions. BRs are a useful aid for plant propagation. For example, treating wooding cuttings enhances the root response. Micropropagation of cassava and pineapple has been improved by BR treatment. In order to complete this section on plant hormones, it might be advisable for you to watch the following YouTube videos. One is a simplified animation of stomata control. The second one is a summary on plant hormones. I hope from this lecture that you have understood about what hormones are, their classification, and the many diverse roles that they have in plants. You should be able to give examples and understand the processes of auxins, gibberellins, cytokinins, ethylene, ascorbic acid, and brasinosteroids. You should also understand that biosynthesis of many of these hormones is complex and requires many intermediates. And finally, that in the role and function, some of these hormones work together. Thank you. This is the end of the lecture.